Welcome back to the Cellular Molecular Biology course. I'm your course instructor Niyatendra. In this video, we will explore peroxisomes, the small yet vital organelles within eukaryotic cells. They play important roles in cellular metabolism, especially in detoxification and lipid metabolism. Now let's see an introduction to peroxisomes. What are peroxisomes? Peroxisomes are small, membrane-bound organelles found in nearly all eukaryotic cells. Although often overshadowed by mitochondria, their roles are important, particularly in breaking down fatty acids and neutralizing toxic substances. In the illustration, you can see the eukaryotic cell in which the peroxisome is highlighted. Now let's see the origin and structure of peroxisomes. Peroxisomes are formed from pre-existing peroxisomes by growth and division. They are surrounded by single membrane and contain oxidative enzymes such as catalase and urate oxidase that facilitate their metabolic task. Peroxisomes can also originate from the endoplasmic reticulum. Unlike mitochondria, they do not carry their own DNA, relying instead on the import of specific proteins synthesized in the cytosol. Structurally, these organelles are enclosed by a single membrane and contain powerful oxidative enzymes concentrated in a dense, sometimes crystalline core. Their compact architecture allows them to perform rapid detoxification and metabolic reactions essential for the cell health. In the illustration, you can see the peroxisome structure with a lipid bilayer. This crystalline core is not always present. Now let's see the key functions of peroxisomes. One of the main functions of peroxisomes is beta oxidation, that is the breakdown of very long chain fatty acids into shorter chains. This process produces acetyl-CoA, which is then transported to mitochondria for ATP production. In the illustration, you can see the fatty acid breakdown, where you can see the beta oxidation, which produces heat, reactive oxygen species, and acetyl-CoA. Peroxisomes contain catalase, an enzyme that converts toxic hydrogen peroxide, which is a byproduct of cellular reactions, into water and oxygen. This protects cells from oxidative stress and potential damage. In the illustration, you can see the ROS metabolism, in which we have seen earlier that the beta oxidation produces acyl-CoA and ROS. One of the reactive oxygen species is the hydrogen peroxide, which is then converted to water and oxygen. with the help of the enzyme catalase. So this ROS metabolism is also carried out by the peroxisomes. Peroxisomes play a central role in lipid metabolism. They are responsible for the initial breakdown of very long chain fatty acids, something which organelles like mitochondria can't perform. Through detoxification, these fats are shortened so that other organelles can handle the rest. The peroxisomes also produce plasmalogens, which are unique phospholipids essential for nerve and heart tissue. Additionally, they help convert cholesterol into bile acids, thus supporting digestion and lipid regulation. This combination of breakdown and synthesis activities makes peroxisomes key players in maintaining lipid balance within the cell. In plant cells, peroxisomes take on additional and vital roles. One key function is photorespiration, where they help convert glycolate, which is a byproduct of photosynthesis, into less harmful compounds, working in coordination with mitochondria and chloroplast. In germinating seeds, special peroxisomes called glyoxysomes convert fatty acids into sugars to fuel early growth. These organelles also detoxify hydrogen peroxide and contribute to the production of essential plant hormones. These roles underline how peroxisomes support growth and energy balance and also stress responses in plants. Now let's see the differences between peroxisomes and lysosomes. While both peroxisomes and lysosomes are membrane-bound organelles, which are involved in cellular cleanup, they serve distinct roles. Peroxisomes specialize in breaking down toxic molecules like hydrogen peroxide and metabolizing fatty acids, while in contrast, lysosomes are packed with digestive enzymes that break down cellular waste, damaged organelles, and invading pathogens. Peroxisomes mainly use oxidative reactions, while lysosomes rely on hydrolytic processes. Though both contribute to cellular homeostasis, their origin and specific enzyme types make them fundamentally different. Now let's see the clinical significance. When peroxisomes malfunction, serious disorders can result. Zellweger syndrome, for example, is a genetic disorder that impairs brain development and metabolic processes due to defective peroxisomes. Another peroxisomal disorder is the X-linked adrenoleukodystrophy. which is also a disorder caused by toxic fatty acid accumulation. Maintaining healthy peroxisome is essential for metabolic balance, preventing neurodegeneration, and supporting immune responses, and also for development, especially in early life. Now let's see research and therapeutic approaches. Research into peroxisomal disorders has grown significantly in recent years, especially as scientists have identified specific gene mutations like those in the PEX gene family that disrupt peroxisome formation. Current experimental therapies include gene therapy aimed at correcting these mutations, as well as enzyme replacement therapy, although the latter remains in early development. Dietary interventions have shown modest success, particularly in managing symptoms of metabolic imbalances. In the laboratory, researchers use model organisms to better understand these complex diseases, while the gene editing tools like CRISPR offer hope for more precise treatments. Despite these advances, challenges remain, especially in treating neurological forms where drugs must cross the blood-brain barrier. The future of peroxisomal research is promising with the potential to significantly improve both diagnosis and therapeutic outcomes. So, peroxisomes are vital organelles involved in breaking down fatty acids, in detoxifying harmful substances, and synthesizing key lipids. While they may be small, their impact on cellular health is profound. In plants, they contribute to metabolic processes like photorespiration, and in animals, they contribute, that is, they collaborate along with mitochondria to maintain energy balance. Disorders from peroxisomal malfunction, such as the Zellweger syndrome, highlight the importance of these organelles. The ongoing research offers hope through novel treatments, including gene therapies and enzyme-based solutions. So, understanding peroxisomes is essential not just for cell biology, but for developing future medical therapies that address complex metabolic disorders. Thank you for watching. See you on the next video.